This is Dr. Morrison. We're going to go ahead and finish the lecture series on Chapter 4. This will be Part B, again, functional anatomy of the prokaryotic and the eukaryotic cell. Now, we were talking about the plasma membrane before we broke. Now, let's talk about the selective permeability, one of the major characteristics of the plasma membrane, which allows the passage of some molecules. Enzymes for ATP production, for example, are allowed to pass. Photosynthetic pigments on folding called chromophores and thicolores, as I referred to earlier. Damage to the membrane by alcohol, quaternary ammonias, detergents, polymix, and antibiotics causes leakage of cell content. Now you may ask, why do we go through all this anatomy of the cell membrane in bacteria? Well, this is one of the reasons why. When we talk about antibiotics later on in the semester, it's important for you to recognize how they work. As you can see, certain antibiotics, such as polymyxin antibiotics, break down the cell membrane, allow leakage of cell contents, obviously allowing the environment that's outside the cell membrane, that inside the cell, obviously will change the environment and thus destroy the cell. You can see certain alcohols and ammonia detergents also cause the same phenomena. Material crosses the cell membrane by passive processes such as simple diffusion, movement of molecules or ions from areas of higher concentration to areas of lower concentration. At equilibrium, the concentration gradient has been eliminated. Osmosis is a definition that you should be familiar with. It's the movement of solvent molecules across selectively permeable membrane. Osmotic pressure is the force which a solvent, such as water, moves from a solution of lower solute concentration, such as in dissolved sugar, to a solution of higher solute concentration. I know most of you are already familiar with this process from AMP1. Isotonic, hypotonic, hypertonic solutions, you should be familiar with each of those, have equal isotonic solutions, have equal solute concentrations of both sides of the cell membrane. Hypotonic solutions have a lower concentration of solute outside the cell membrane than inside. Hypertonic solutions have a higher concentration of solute outside the cell, and bacterial cells placed in such solution lose water by osmosis and will eventually shrink. Cytoplasm will collapse, and thus will the cell wall. I'll give you an example of that. If you have ever had a dental procedure done, and the dentist will tell you to gargle with salt water. Gargling with salt water, which is a hypertonic solution, will kill the bacteria inside the mouth. Why? The process of osmosis will allow water to leave out of the bacterial cells. The cell wall will collapse and the bacteria will be destroyed. So the use of a hypertonic solution, a hypertonic sodium solution, would be effective in killing bacteria. Simple diffusion, again, the term you should already be familiar with, movement of solute from an area of high concentration to an area of low concentration. And then facilitative diffusion, solute combines with a transporter protein, so the protein actually facilitates the transmission or the passage of the solute in the membrane. Movement across the cell membrane, again, this is review for most of you from AMP1. Movement of substances across the cell membrane can be facilitated by the use of a protein channel, allows the substances in, as you can see, as it enters into the channel and then released inside the cell. Osmosis, as we already described, movement of water across a selectively permeable membrane from an area of high concentration to an area of low concentration. Osmosis. Osmotic pressure, pressure needed to stop the movement of water across the membrane. Again, this looks at what happens to the cell in the environment of an isotonic solution where the solute concentration is equal on both sides, a hypotonic solution where the concentration outside the cell of solute is lower than inside the cell. Fluid would tend to move inside. Water moves into the cell. Notice the water moves into the cell here, and then here the concentration of fluid in a hypertonic solution outside the cell more concentrated. Water would move outside the cell to this area, causing a collapse of the cell wall. Now the risk in the hypotonic solution that I should have mentioned is that water, as it rushes into the cell, puts an enormous strain on the plasma membrane and would rupture the cell, causing osmotic lysis. So hypotonic solutions in some circumstances can destroy bacteria as well. Movement across the membrane. Active transport of substances requires a transporter protein, as showed to you in a previous illustration, and it requires energy in the form of ATP. Group translocation. Substances require a transporter protein to be effective. The term cytoplasm refers to everything inside the plasma membrane. It has many inclusions, such as metachromatic granules that are stored in phosphate, volutin, polysaccharide granules of glycogen and starch, and lipid inclusions such as polybeta hydroxybutyric acid and sulfur granules. Cytoplasm also contains many ribosomes, the site of protein synthesis. Again, you should be familiar with that with AMP1. Carboxysomes, which are inclusions found in bacteria that use carbon dioxide as the sole source of carbon, and then gas vacuoles or gas vesicles help some bacteria maintain buoyancy. Bacterial chromosomes, which contain genetic information, is a single long circular molecule of DNA found in the nuclear area of nucleoids. Small circular DNA molecule plasmids are not connected to the chromosomes and replicate independently. Plasmids do not contain normal essential genes, but may provide a selective advantage under abnormal conditions, antibiotic resistance for example. Imagnosomes are inclusions of iron oxide formed by few gram negative bacteria that aid microbes in orienting itself environmentally. The cytoplasm, just in a review, substances inside the plasma membrane. Here, jelly-like substance. Nuclear area or nucleoid inside the nucleus. Ribosomes. Ribosomes, again, inside the cytoplasm, important in protein synthesis. Some of the inclusion bodies that we see inside the cell: metachromatic granules, polysaccharide granules, lipid inclusions, sulfur inclusions, carboxysomes, gas vacuoles, and magnosomes. And you can see what the constituents in are for here and what their available and role are. Phosphor reserves, energy reserves. We already talked about the protein, silver, and iron oxide in magnosomes. Endospores. Endospores are important structures that you should be familiar with as well. Again, those of you who are already into your lab sections, and lab 2, I believe, goes into some cell coverings, and endospores are described specifically. Endospores are highly resistant bodies formed by few bacterial species such as Bacillus and Clostridium. I think in your lab we talk about Clostridium tetani as having a endospore. Sporulation or sporogenesis is a process of their formation. First, there's an ingrowth of the plasma membrane, a spore septum. A small portion of the cytoplasm and a newly replicated bacterial chromosome is then surrounded by a membrane and a forespore. A thick spore coat of protein forms around the membrane. The endospore core is dehydrated and contains considerable dipolylglycolic acid, as well as few essential materials necessary to return it to a vegetative state, which is accomplished through a process called germination. Resting cells resistant to desiccation, heat, chemical, Bacillus, Clostridium, sporulation, endospore formation, germination, the return to a vegetative state. This is a picture of an endospore. In eukaryotic cells, when comparing prokaryotic and eukaryotic cells, prokaryotic cells again come from that Greek word prenucleus, as we described earlier, and eukaryotic cells come from this term true nucleus. Flagella and cilia, we've already talked about flagella. In the eukaryotic, flagella are relatively long. Cilia are more numerous and typically are shorter. Cilia you may be familiar with when we talked about the respiratory system in AMP1 and in AMP2. We talked about cilia is the little tiny hair-like follicles that actually will move substances in the cell through a process of locomotion. Both are involved in locomotion, both cilia and flagella, and both contain small tubules of proteins referred to as microtubules. Example of flagella, as you see on the left, and cilia, as you see on the right. Microtubules, the arrangement of the tubulin in these cells, 9 pairs plus 2 in the arrangement. You've already reviewed this in AMP1. Most algae and some fungi have cell walls. We talked about the glycocalyx and the cell walls already, but as you can see in most algae, the arrangement is somewhat different. Fungi have cell walls containing cellulose. Often fungi have chitin as well. Yeast cell walls contain polysaccharide glucan and mannan. No eukaryotic cell wall contains peptidoglycans. Protozoa have flexible outer cell coverings called pellicles. In animal cells, the plasma membrane is covered by a sticky carbohydrate covering referred to as the glycocalyx. As you look at the cell wall, plant, algae, and fungi, and carbohydrate in the cell wall, cellulose, chitin, glucan, mannan. Glycocalyx, which we've already described, is the carbohydrate covering extending from the animal plasma membrane, bonded to proteins and lipids in the membrane. In eukaryotic cells, the plasma membrane, which contains sterols, may be external cell covering. Substances cross the membrane by mechanisms similar to that of prokaryotes. In addition, a process of engulfment, endocytosis, brings particles, even some viruses, into the cell. Examples are phagocytosis, used by white blood cells to engulf and destroy bacteria, and pinocytosis, by which liquids and dissolved substances enter the cell. Again, we've described all of these processes in AMP1. The plasma membrane, this is a review as we described already earlier. In the eukaryotic cell, phospholipid bilayer has peripheral proteins and enteral proteins, also transmembrane proteins. Sterols also exist in the cell membrane, and then the glycocalyx of carbohydrates. The plasma membrane is selectively permeable. Also, the processes, again, that we've already discussed: simple diffusion, facilitated diffusion, osmosis, active transport, and endocytosis. You should be very familiar with each of the definitions for those terms. Phagocytosis, pseudopods extend and engulf particles. Pinocytosis, the membrane itself actually folds inward, bringing fluid in and dissolved substances. In the eukaryotic cell, again, the cytoplasma membrane, substances inside the plasma and outside the nucleus. It's where the cytoplasm would be located. Cytosol is the fluid portion of the cytoplasm. The cytoskeleton is the microfilaments and intermediate filaments and microtubules. And the cytoplasmic streaming is the movement of cytoplasm throughout the cell. I know all of these terms you're familiar with from AMP1, but again, you can apply this now in microbiology as we describe the eukaryotic cell. And again, you should be familiar with each of these terms and what they refer to. Cytoplasma. Cytoplasma is the matrix which various cellular components are found. Complex of internal structures and microtubules and intermediate filaments and microtubules are the cytoskeleton. Movement of cytoplasm from one part of the cell to another is referred to as cytoplasmic streaming. In eukaryotes, unlike prokaryotes, many important enzymes are found in and functions are carried out by organelles. The nucleus is an oval organelle containing DNA. It is surrounded by a nuclear envelope. The nucleopores in the nuclear membrane allow the nucleus to communicate with the endoplasmic reticulum of the cytoplasm. The nucleoplasm is a gel-like fluid in the nucleus. The nucleoli may be in the center, and the synthesis of ribosome RNA are present. DNA is combined with protein histones and non-histones inside the nucleus. The combination is referred to as the nucleosome. When the cell is not reproducing, DNA and associated proteins are visible as a mass called chromatin. When reproducing, chromatin becomes visible as rod-like bodies referred to as chromosomes. This is an electron microscopy of the nucleus. You also can see the nucleolus. This illustration easily typifies the structure of the nucleus and the nucleolus. Endoplasmic reticulum and ribosomes. Again, these are areas that were described in AMP1, so it should be a review for most of you. Within the cytoplasm, there's a network of flattened sacs or cistern referred to as the endoplasmic reticulum. It functions to synthesize and store lipids and proteins and transports them. The end of the cistern pinches off into secretory vesicles which transport substances within the cell. The rough endoplasmic reticulum has ribosomes bound to it, and the smooth endoplasmic reticulum does not. Ribosomes are the site of protein synthesis in cells. This is an illustration of the endoplasmic reticulum, as you can see it in electron microscopy. And then, as you can see, remember, smooth endoplasmic reticulum lacks the ribosomes. The rough endoplasmic reticulum has the ribosomes, the site of protein synthesis. Smooth endoplasmic reticulum is involved in carbohydrate production, whereas you see the rough endoplasmic reticulum is involved in protein because of the presence of ribosomes. The Golgi complex consists of a network of flattened sacs called cistern, which, again, as described in AMP1, looks like a stack of dishes. Like the endoplasmic reticulum, these functions to export substances from the cell and transport substances within it. The Golgi complex receives proteins and lipids from the endoplasmic reticulum and delivers them to secretory vesicles. The Golgi complex mitochondria and organelles with smooth outer membranes and inner membranes arranged in series of folds called cistern or cristae. Semi-fluid center of the mitochondrion are called the matrix. Enzymes forming ATP are located on the cistern, which is one reason why mitochondria are called the powerhouse of the cell. This is the mitochondria. These are the matrix, the cristae, the inner membrane, and the outer membrane as described. Chloroplasts. These are photosynthesizing cells, contain membrane-bound structures called chloroplasts, which contain chlorophyll and enzymes involved in photosynthesis. Now note, it contains both chlorophyll and the enzymes involved in photosynthesis. Chlorophyll is found in a membrane called thylakoids, which we referred to earlier. Stacks of thylakoids are called grana. This is a cell with the granulum or the granum. Now, lysosomes. White blood cells which destroy bacteria by phagocytosis contain lysosomes. The membrane-enclosed spheres contain powerful digestive enzymes. This illustration demonstrates to you the lysosomes inside the cell. A vacuole is a space or a cavity in the cytoplasm that serves as a storage for storage and other functions. Again, electron microscopy depicting the vacuoles are the empty spaces inside of the cell. Peroxisomes are organelles similar in structure to lysosome but smaller. They also contain enzymes. They oxidize various organic substances. The enzymes catalyze decompose toxic hydrogen peroxide inside of cells. Centrosomes. The centrosomes consist of a pericentriola area, which is organizing center for mitotic spindle and plays a critical role in cell division. Within these areas are located centrioles, arrays of microtubules that play a role in the formation and regeneration of cilia and flagella. Centrioles are bundles of microtubules that probably play a role in cell division. These are the organelles, as I've already described. I would familiarize yourself with this slide, and the previous information I described tells you the components and the functions of many of the organelles inside the cell. In eukaryotic cells, there are certain structures that are not membrane bound: ribosomes, centriosomes, and centrioles. Ribosomes, which are involved in protein synthesis. Membrane bound, they're free. They come in different sizes, 80S and 70S. I didn't focus a lot of attention on this, but you just should be familiar with ribosomes and what their roles are. And then the evolution of eukaryotes. There's a theory that explains the origin of eukaryotes from prokaryotes. This was pioneered by Lynn Margulis. It's referred to as the endosymbiotic theory. Should be familiar with this theory. Larger bacterial prokaryote cells are presumed to have engulfed smaller bacterial cells. One organism living within another basically is called endosymbiosis, and eventually evolved into eukaryotic cells. Mitochondria and chloroplasts in eukaryotic cells are considered evident of this theory. They resemble prokaryotic cells and can reproduce independently of their eukaryotic host. Simply stated, the theory of endosymbiosis is the concept that mitochondria and chloroplast are the results of years of evolution initiated by endocytosis of bacteria and blue-green algae, which instead of becoming digested became symbiotic. Illustration of the theory.